Welcome to this explainer. Today, we're focusing our attention on the ultimate engine of existence. We're talking about the biological machinery that literally sustains every living thing on Earth. It's a process we've all heard of, right? But honestly, few of us truly understand it at a deep mechanical level. So, we're going to unpack the incredibly elegant physicochemical process of photosynthesis.
Here's our roadmap for today. We'll kick things off with the macro-level stakes of survival in the basis of life, then trace the history in early scientific experiments. After that, we'll step inside the chloroplast, analyze the colors of photosynthesis, and finally get down to the actual quantum physics in the light reaction. Let's get into it.
Section 1. The basis of life. Let's establish the massive stakes of our topic right off the bat.
Have you ever wondered where plants actually get their food? It sounds like a child's question, doesn't it? We all know animals eat plants or, other animals. But what sustains the foundation of all of that? Most of us just kind of take for granted that green plants just grow. But actually, they're operating a highly advanced manufacturing process. You see, the entire living world is basically divided into two camps. First, you have autotrophs, like green plants, which literally synthesize their own food using nothing but light energy. And then you have heterotrophs. That includes all animals, and us humans too. We depend entirely on those plants. Ultimately, every living form on Earth depends on sunlight for energy. This physicochemical process we call photosynthesis, it's the primary source of all food on Earth, and honestly, it's the sole reason we even have an oxygen-rich atmosphere to breathe.
Moving right along to Section 2, Early Scientific Experiments. This whole mechanism is entirely invisible to the naked eye, so how did we even figure it out?
It's a fascinating, historical detective story, really. Science is all about passing the baton. Back in the 1770s, Joseph Priestley revealed the essential role of air. By 1779, Jan Ingenhousz proved that sunlight is absolutely essential to the process. Jumping ahead to 1854, Julius von Sachs discovered that plants produce and store glucose. And then, in the 1880s, T.W. Engelmann literally split light with a prism to figure out which specific colors of the spectrum actually drive this reaction. Let's think about Priestley's genius for a second with his 1770 bell jar experiment. He placed a burning candle and a live mouse inside a closed glass bell jar. Now, as you might guess, very quickly the candle went out and the mouse suffocated. He realized that breathing animals and burning candles somehow, quote-unquote, damage the air. But, and here's the kicker, when he placed a live mint plant in that exact same jar, the mouse survived and the candle could keep burning. Priestley proved that plants actually restore to the air whatever animals and candles take out, a real game changer. But the absolutely crucial point in our modern understanding actually came much later, from a microbiologist named Cornelius von Neal. By studying bacteria, he demonstrated that photosynthesis is essentially a light-dependent reaction where hydrogen reduces carbon dioxide into carbohydrates. More importantly, he proved a totally mind-bending fact. The oxygen that green plants release into the air, it doesn't come from the carbon dioxide they take in. No way. It actually comes from the plant literally splitting water molecules, H2O, using the immense power of sunlight.
Okay, Section 3, Inside the Chloroplast. Now we're zooming in from the whole plant, past the leaf, right down to the specific cellular factory where the magic actually happens. If you were to crack open a mesophyll cell inside a leaf, you'd find these incredible structures called chloroplasts, and the architecture inside is just brilliant. There's a very clear division of labor. You have a membranous system made up of grana and stroma lamellae, and surrounding all of that, you have a fluid matrix called the stroma. The plant perfectly aligns these flat chloroplasts right against the cell walls to catch the absolute optimum quantity of incoming light. It's like they're setting up tiny solar panels. Within that architecture, there's a strict, highly efficient division of labor. The membranous system is responsible for the light reactions. It physically traps light energy and creates high-energy chemical intermediates, specifically ATP and NADPH. Then out in the fluid stroma, the dark reactions take place. Now, actually, scratch that. Don't let the name fool you. They don't only happen in the dark. It just means these enzymatic reactions aren't directly driven by light. Instead, they depend entirely on the ATP and NADPH generated by the light reactions to actually synthesize the sugar. Which brings us to Section 4, the colors of photosynthesis. So how does that membrane actually catch the light in the first place?
Well, if you've ever wondered why there are so many different shades of green out in nature, it's because leaves use a whole mixture of tools to catch light.
If you separate leaf pigments using paper chromatography, you don't just see one uniform green. You actually find 4 distinct pigments. There's chlorophyll A, which is a bright blue-green, chlorophyll B, a yellow-green, xanthophylls, which are yellow, and carotenoids, which are yellow-orange. What's truly fascinating is how these pigments overlap. The specific wavelengths of light that chlorophyll A absorbs best, which peaks right in the blue and red regions of the spectrum, align perfectly with the peak rate of photosynthesis. It is unequivocally the chief pigment driving this entire engine. It's doing the heavy lifting. So what do the other 3 pigments even do? Well, we call them accessory pigments. They're like crucial sidekicks. They absorb different wavelengths of light and then transfer that energy over to chlorophyll A. This drastically broadens the usable spectrum of light the plant can harvest. But even more importantly, they protect that vital chlorophyll A molecule from being destroyed by photooxidation when the sunlight gets just a little too intense. Let's move to our final section, Section 5, the light reaction. It's time to zoom all the way into the micro level and look at the actual quantum physics of this photochemical phase. To trap light efficiently, those pigments we just talked about don't just float around randomly. Not at all. They are meticulously organized into what we call a light harvesting complex, or LHC. Think of it as a massive, highly efficient molecular satellite dish. Each complex is made of hundreds of pigment molecules bound tightly to proteins, all acting together as a giant antenna system. Just imagine that satellite dish in action. A photon of light strikes one of the outer pigment molecules. Bam! That energy is then rapidly bounced inward, from molecule to molecule, funneling deeper and deeper until it hits a single, specialized chlorophyll A molecule right in the very middle. We call that the reaction center. Now, within the plant's membrane, there are actually 2 distinct types of these satellite dishes, aptly named photosystem 1 and photosystem 2. And a quick side note, they're actually named in the order they were discovered, not the order they operate in. Kind of confusing, I know. But the reaction center in photosystem 1 has an absorption peak at 700 nanometers, so we call it P700. Photosystem 2 peaks at 680 nanometers, so you guessed it, it's called P680. The really crucial point is what happens the exact millisecond light hits the system. When photosystem 2's reaction center absorbs red light, something amazing happens at the atomic level. An electron gets so incredibly energized by that light that it physically jumps into a farther orbit, away from its atomic nucleus. It is literally bursting with trapped solar energy. And before it can even fall back down, a specialized molecule called a primary electron acceptor reaches in and grabs this excited electron, kicking off a downhill movement known as the electron transport chain. Which leaves us right at the precipice of the greatest chemical relay race on Earth. We've got this highly energized electron trapped from a sunbeam, ready to be put to work. But where do these energized jumping electrons go? How are they channeled through cytochromes? And how do they ultimately build the physical glucose molecules that sustain every single one of us? Well, that complex, beautiful chain reaction is a profound story for another time. But I really hope this explainer leaves you looking at every green leaf you pass just a little bit differently.